Thank you very much, Marcus. And since I can moderate myself, I will allow myself 3 or 4 hours for this presentation. Just kidding. That's a joke. Okay. Thanks very much. It's great to be here with everyone, even in virtual space. I am going to go over the basic biology and genetics of GIST. Some of this material will indeed duplicate what you've already heard, but I think you will be hearing it from a somewhat different perspective. I'm not a physician, I'm a scientist, so it's more of a scientific than a medical overview, although of course the science and the medicine are closely interrelated. So today, this morning where I am, I want to tell you a little bit about where GISTs come from, and we'll talk about the difference between carcinomas and sarcomas. And then I want to give you some background about the molecular biology. You've already heard reference to KIT as a protein and as a gene, so I want to go over in a little more detail what you've already heard about IHC, or immunohistochemistry, and mutational testing. And again, I'll say a little bit more about TKI drugs. So all of these topics have been touched upon already this morning by our speaker from Holland, but we'll be looking at them from a somewhat different angle. So let's begin by thinking about what GIST is as a disease. It is a form of cancer. Cancers can begin in almost any type of cell in the body. The human body is comprised of many thousands of different kinds of cells. And this is a fundamental and important point to understand, to appreciate, that what really counts in understanding a cancer is to know the type of cell from which it began. We need to know the type of cell from which a tumor developed. That's essential knowledge if we want to understand the best treatment of the cancer. And that's true independent of the organ in which the tumor develops. It's the cell type, not the organ, that matters most. To give two examples of that, we often hear reference to skin cancer, which is a very common cancer, but the common skin cancer is a basal cell carcinoma, as it's called. There's also, for example, a melanoma. That's also a kind of skin cancer, but it's a completely different disease from basal cell carcinoma. Or if you think about lung cancer, another common cancer, the most common lung cancer is the adenocarcinoma, but there's also, for example, mesothelioma, often caused by asbestos exposure. And again, those are completely different diseases. They originate in different cell types, and their treatment and prognosis are completely different. GIST is a sarcoma rather than a carcinoma. The most common cancers that we hear about are carcinomas. These are cancers that arise in the epithelial tissues, as we say. That is, the tissues that the cells that line the interior or exterior linings of the body. That includes, for example, the skin, the breast, the colon, which is the external surface, even though it's inside the body. It's the external surface of the gastrointestinal tract. Lung, stomach, and many other common cancers. Sarcomas, in contrast, are cancers that arise in connective or supportive tissues. Some examples you may have heard about are osteosarcoma, osteo for bone, liposarcoma, lipo for fat, angiosarcoma, angio for blood vessels. So these are cancers that arise in connective or supportive tissues like the fat, the blood, the bone, the muscle. Sarcomas are relatively rare as human cancers. Only about 1% of adult human cancers are sarcomas. That's why sarcomas are generally treated by specialist oncologists, typically in a sarcoma center of excellence, more so than by general oncologists. By the way, sarcomas, although they're rare in humans, they're actually quite common in, for example, dogs. They're much more commonly treated they're much more frequent at a veterinary clinic than in a human hospital, but actually quite common in canines. And in fact, the dog is often an important model system for studying sarcoma. We heard reference this morning to people. We defined a dinosaur as someone who has attended, what, more than 12 New Horizons meetings? But in fact, what I'm showing you here is a real dinosaur. This is a recent medical diagnosis of an osteosarcoma. It's one of the few tumors that one can observe in a fossil. This diagnosis was actually made in Canada. Unfortunately, the diagnosis came too late for the patient. The patient died 77 million years ago, possibly from the osteosarcoma. We don't know, but this demonstrates that sarcoma is not a new disease. In fact, it's been with us for many millions of years. Now, to compare carcinomas versus sarcomas in the context of the gastrointestinal tract, where a GIST begins, the common stomach or intestinal or colon cancers, as I said earlier, are carcinomas. These begin in the epithelial lining of the gut, inside the gut, but therefore outside, topologically outside the human body. They're the outer surface, the inside surface of the gut, therefore, if that's clear. And what I'm showing in the top left is a cross-section of the gut. The innermost core part is really the external surface. That's where the food, that's where the food goes through the gut, the lumen of the gut, as we say. And I've indicated with the yellow star that this is where carcinomas begin, colon or stomach cancers, in the lining epithelial surface. There's a larger blow-up picture in the main part of the slide. And again, you can see the epithelial cells lining the so-called villi of the intestine, the invaginations in the in the center of the intestine. That's where the carcinomas begin, like a common colon cancer. But in fact, GISTs begin not there, but in the muscular wall that surrounds the GI tract, as indicated by the red star. So as a sarcoma, it's beginning not in the epithelial tissue, but in the muscular wall. And that's apparent to the doctor who is examining the tumor by endoscopy or by a CAT scan, and it's certainly evident at the time of surgery. Now, you heard reference earlier to peristalsis. There is some importance of understanding the relationship between peristalsis and GIST. So what is peristalsis? When you digest food, the food is actively pushed through the gastrointestinal tract. It doesn't just fall, but it's pushed. And it's pushed by waves of muscular contraction, coordinated waves of squeezing of the GI tract by the surrounding muscle. Those waves are called peristalsis. Peristalsis meaning these coordinated waves of contraction. And as shown in the picture, boluses or pieces of food are pushed all the way through the gastrointestinal tract by these waves, contracting waves of muscular contraction. Well, that has to be coordinated. The muscles have to do this in a synchronized, coordinated way. Just as when we have a rowing crew at the Olympics, as shown in the picture at the bottom, all the oars have to be coordinated and put into the water at the same time. People have to coordinate their strokes. In fact, there's a special little person at the front of the boat called the coxswain, whose job is to call out stroke, 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 to keep all the muscles coordinated. So you need a coordinating person. That coordinating person, in the case of gastrointestinal peristalsis, is a particular neuromuscular cell. It's a particular kind of cell called the ICC, the interstitial cell of Cajal. These are the cells that send out the electrical pulses, effectively the signals, that cause the muscle in the GI tract to contract. The interstitial cells of Cajal, we call these the pacemaker cells. They're the coordinators of gastrointestinal peristalsis. The picture in the middle is just a trace of the electrical activity, so-called membrane potential, the electrical activity of an actual functioning set of interstitial cells. In fact, this was done in the mouse, and you can see that over every few seconds in the mouse, there's a rhythmical change of electrical potential, which drives the muscles to contract. So the interstitial cells of Cajal, these cells were first identified by a famous Spanish histologist named Cajal, shown in the stamp in the picture. These cells, which were identified by Cajal, are found throughout the gastrointestinal tract, and there's a diagram on the left that just shows you how these ICCs are distributed. In fact, in several layers. It's a little more complicated than shown here, but essentially they're distributed around within the muscle surrounding the gastrointestinal tract, all the way along the GI tract. So now we can understand why, since the ICC is present throughout the GI tract, and since GISTs arise in the ICCs, or at least in closely related precursor cells, that explains then right away why gastrointestinal stromal tumors can arise anywhere along the GI tract, as shown here, all the way from the esophagus to the rectum. Most commonly in the stomach, but they can arise anywhere along the GI tract. So these are all GISTs. They may have a little bit different biology depending on where they arise, but they're all GISTs and they all arise in the interstitial cells. Let's now talk about metastasis. GISTs, like any other cancer, has the potential to spread throughout the body. That is, cells can be shed from the initial primary tumor. They can travel through the body or through the bloodstream and move to other locations. When a GIST is first diagnosed medically in a patient, it may be localized. It may still be a single primary lesion somewhere in the GI tract, or in many cases, it is not diagnosed until it has already metastasized, either locally in the peritoneum or to other organs, notably the liver. A very important take-home message from this presentation, which I hope everyone takes to heart. A GIST metastasis is still a GIST, and it has to be treated as a GIST. It is, even if a GIST cell has migrated to, say, the liver or the lung or the bone and has begun to grow again, it has not become a liver cancer. It's still a GIST. And I'm showing you a record by the musician Sting. He's an Englishman, and he recorded a song called Englishman in New York. And the point of this is simply, when an Englishman moves to New York to play a concert, he's still an Englishman. He hasn't become a New Yorker. A New Yorker like Norman Scherzer will instantly recognize that Sting is not from New York. So just in the same way, when a GIST cell has moved into a different organ like the liver or the lung, it's still a GIST cell. Now I want to spend a few minutes talking about the molecular biology of GIST. You've already heard some reference to this in the first presentation. But I want to look at this from the point of view of a molecular biologist or scientist. So you will hear talk about proteins and about genes and about the relationship between proteins and genes. Proteins, as shown in the curly diagram on the left, are the molecules that do most of the work in the body structurally and functionally, functioning as catalysts in metabolism and regulating signal transduction. Genes, as illustrated by the DNA model on the right, genes, DNA, are the blueprints that encode the instructions that the body uses to make proteins. So for every protein, there is a corresponding gene. And for every gene, there is a corresponding protein, sometimes even more than one protein. So what are proteins chemically? Proteins are polymers made by linking together building blocks, subunits, if you like, Lego bricks assembled into a Lego structure. And those subunits are called amino acids. There are 20 types of amino acids found in proteins. Each one has a historical name, and each one can be represented by a single letter in the alphabet. Since we have 26 letters, at least in the English alphabet, and there are 20 types of amino acid, we can assign one letter to each amino acid, and we do. So an A, for example, represents the amino acid alanine, D represents the amino acid aspartic acid, and so on. How long are proteins? Proteins can be anywhere from really just a few dozen up to tens of thousands of amino acids in length, depending on the protein. As we just said then, genes, DNA, the genome, are the codes that encode, they are the blueprints, if you will, that encode the sequences of proteins. The human genome encodes at least 30,000 different proteins. KIT is one of them, PDGFRA is one of them, and so on. Oncogenes. Oncogenes were discovered by basic science research, really starting by looking at tumor viruses back in the 1970s. An oncogene then is a human gene which, when it is mutated, can drive the uncontrolled growth of a cancer cell. So a mutation occurring in an oncogene can initiate a tumor. That fundamental work was recognized by the Nobel Prize back in 1989 to Bishop and Varmus. Even from the beginning of that research, even from the time of that discovery, it was already apparent to some people that an oncogene therefore represented a potential target for cancer treatment. And the protein product of an oncogene is a potential target for cancer treatment. It took more than 20 years to turn that idea into reality. And in fact, imatinib was one of the very first drugs that fulfilled that promise of taking the concept of an oncogene and turning it into the basis of a cancer treatment. The first great success was obtained with the treatment of CML, which we heard referred to earlier, chronic myelogenous leukemia. And very shortly afterwards, the same drug, imatinib, was tested in GIST and had the remarkable beneficial therapeutic effect that we saw discussed in the first talk. Just to give a little historical background then, this is Brian Drucker of Oregon Health Sciences University, probably the single most important medical pioneer in the development of imatinib. The drug itself, imatinib, is shown on the right. Initially, as you saw in the first talk, it was just had the telephone number STI 571. Then it had the brand name Gleevec, developed by Novartis Pharmaceuticals. In fact, it was developed as a treatment for chronic myelogenous leukemia because it inhibits the oncogene that drives CML. the so-called ABL oncogene. And the initial paper shown up at the top of the slide, targeting ABL as a therapy for CML. But in fact, it was shown shortly after that it was discovered to also effectively inhibit KIT, the oncogene that drives GIST, and therefore to be an effective therapy for GIST. Okay, everything changed in 1998. Between 1998 and 2000 was the turning point for the understanding of GIST. It was also the time when my sister-in-law was diagnosed with GIST and when I first became interested in the subject. That was the turning point in the development of therapy for GIST. In fact, the critical discovery was published in 1998. We will see that in just a minute. The breakthrough about the role of the KIT oncogene in GIST. Before we get to that, I'll just say a few words about the background of KIT. You've heard so much about KIT already, and you'll hear a lot more during the remainder of the conference. People sometimes wonder why it's called KIT. Actually, this gene was originally discovered back in the 1980s as a gene found on a particular virus, a virus that infects and causes a cancer, a sarcoma, in cats. And so it was named KIT precisely for KIT as a bit of a, maybe a little bit of a joke, but to refer to the feline origin of this virus. So it really is KIT for kitten or cat. After that initial discovery of the KIT gene, there was a lot of basic research, scientific research done on KIT and its role in the development of various cell types, notably blood cell types. In fact, a lot of that work, I'll just acknowledge, was done very close to where I am now. It was done in Toronto by Alan Bernstein and his colleagues, who did a lot of the early work characterizing the biology of the KIT gene and the KIT protein. So as usual, it's a lot of basic research that leads ultimately to the biological medical application. So this is the turning point. 1998, this publication of this paper, gain-of-function mutations of c-KIT in human GIST. This work was done in Japan by these people whom we should acknowledge with gratitude every time we think about GIST. Hirota Kitamura and their colleagues in Osaka, Japan. The people who put the pieces of the puzzle together and recognized the connection between the KIT gene and GIST. And that completely revolutionized GIST diagnosis and treatment to the point that any research done on GIST before this time is now obsolete. Here are, I think, the two major take-home messages about the breakthrough discovery. First, it was discovered that GIST cells almost always express this particular protein called KIT. We heard mention in the first talk that there are other cell types in the body that express KIT. There are several, but only a limited number, and generally other tumor types do not express KIT. So it is a relatively specific marker for GIST. Secondly, that in most, although not all, but in most GISTs, the gene encoding the KIT protein, the KIT oncogene, has, as is common with oncogenes, has undergone a mutation, a change in sequence. That change in sequence produces an aberrant or different form of KIT, and that aberrant form of KIT drives cell division and therefore drives the development of the GIST cancer. Maybe there are undoubtedly there are other factors as well, but this is a critical factor. And you've seen this picture already, the remarkable early attempt to, the extraordinary decision to use imatinib in a completely unexpected context to treat this rare sarcoma called GIST. And the effect was so immediate and so dramatic that this single patient study of the single patient was published in one of the world's leading medical journals, the New England Journal of Medicine. And I believe at the time, maybe even still today, this is the only time in the history of that journal when a study of a single patient was published as an article, because it was considered such a breakthrough. And with the success in CML and in GIST, Gleevec became a famous drug very quickly. It made the cover of major magazine, Time magazine in the USA. It became the subject of a book called Magic Cancer Bullet. Personally, I'm not sure that I like the metaphor of the bullet, and I don't really like the word magic. It's not magic, it's science, not magic. And it's actually a little dangerous to talk about magic bullets, because if you talk about magic, people expect magical results. The results were dramatic, but it's not magic, it's science. And there are, like any form of therapy, there are limitations to it too. So I'm not particularly in favor of the use of the word magic, but that's the word that's been used, and it does represent the fact that Gleevec was an incredible breakthrough. Okay, back to the science a little bit then. We've heard some discussion of KIT protein in the first talk, and I'll refer to that again. You will sometimes hear KIT called c-KIT. That just means cellular KIT. It's just a synonym. It's the same thing. And you'll sometimes hear it referred to by a kind of telephone number code, CD117. But again, that's just a synonym. It's exactly the same thing. It's KIT, even if you call it something different. So the KIT protein is a particular protein. It is made, or as biologists say, it is expressed by only a few types of adult cells, including the interstitial cells of Cajal, and including almost all GISTs. Dr. Gelderblum referred to immunohistochemistry. That's a long word. If you got that word in Scrabble, you'd get a lot of points. Immunohistochemistry. So what does that mean? IHC. Immuno means that we use antibodies in the technique. Histo means referring to tissues. Chemistry, chemical reactions. So IHC is a process of using antibody chemistry to study tissues. IHC is what the most pathologists working in hospitals are doing every day. They're doing IHC studies, testing tumor samples, particularly by immunohistochemistry. So this is the essential step in diagnosing GIST, to take a sample of the tumor, whether obtained at biopsy or at surgery, and to do this IHC test. So what is the test? We use a very specific reagent. It's an antibody. It's an antibody that recognizes and reacts with, binds to the KIT protein. And so when we treat the tissue, a slice of the tissue on a microscope slide with that antibody, and then do some other special chemical reactions to make the antibody, the presence of the antibody, visible, to visualize the antibody. And we visualize it in the form of a brown stain, a chemical stain. And again, you saw a picture like this before. You see one again. This is an image of a tissue slice that's been stained with that antibody. If the cells stain brown, then that tells us that they express KIT, and that tells us that they are almost certainly GIST. It's a critical part of diagnosis. Here's the example from an actual surgical pathology report obtained by one of our patients. And if you look at your pathology report, and I urge every patient to get a copy of the report, somewhere in the report it will say something like this: immunohistochemical studies, KIT, CD117 positive, that means it did stain, DOG1, which we heard referred to earlier, and it's got another name, ANO1, positive. So those are important results, and it also says other tests. So some other staining tests were done for various other proteins. We don't need to worry about them too much. Proteins like CD34 and desmin. So what do we conclude from that report? The immunohistochemistry tells us that the tumor expresses KIT and DOG1, and that tells us that the tumor is almost certainly a GIST. Also, confirming that diagnosis, we see that the pathologist sees that the tumor does not express various other proteins like desmin that are typically expressed by other sarcomas that might be confused with GIST. So this additional staining confirms that it really is a GIST. Now an important point, the immunohistochemistry only recognizes the protein. It does not distinguish between the normal KIT protein, the so-called wild type, to use the genetic terminology, the normal or wild type KIT, and the mutated forms, the many different mutated forms of KIT. Those are not distinguished by IHC. All the IHC says is that KIT is there. It doesn't tell us what kind of KIT. So I put together this slide, which I won't go through in complete detail, but you can look at it again later. It's a comparison between these two kinds of tests. The immunohistochemistry test that I just talked about, and mutational testing, which we've already had a reference to and we'll say a little bit more about during the rest of the talk and the rest of the conference. It's very important to understand, because there's often confusion about this, that the IHC test and the mutational test are different tests, completely different tests. They ask different questions and they provide different answers. So let's compare on the left column the immunohistochemistry and on the right column the mutational testing, which we will hear more about later. So the IHC stains for the KIT protein using an antibody. The mutational testing is done by DNA sequencing of the KIT gene and other genes. So in the first row of the table, the IHC test tells us whether the tumor cell expresses the KIT protein, and that tells us whether the tumor is a GIST. Often that's just a confirmation of what the surgeon already suspected. On the other hand, the mutational testing determines whether there's a mutation in the KIT gene, in the DNA of the tumor cells, and that will tell us whether the tumor is a KIT-mutant GIST, which is a particular subcategory of GIST. And if so, it'll identify the mutation. To do the IHC, we need a sample of the tumor, typically taken at biopsy or surgery, and the same is true of the mutational testing. We need a sample of the tumor. Often this can be done on a preserved sample that is stored in the pathology lab, so-called formalin-fixed paraffin-embedded tissue. Maybe the last row here is the most important. When is this test performed? Every pathology lab receiving a suspected GIST must do the IHC test for KIT. They'd be negligent if they did not. So that test is always going to be performed. If it hasn't been, that's really borderline malpractice, I would say. So that's always going to be done. Mutational testing. It's not always done. We've already had a poll and vote about that at the beginning of the conference. It's sometimes done, sometimes not. We strongly recommend, for reasons that will become clearer throughout the conference, we strongly recommend that it be done. Often that requires the patient to push and pressure and press to make sure that it is done. Now, I already talked about the 2000 breakthrough of the GIST-KIT connection. That was 1998, 1999, 2000 when that work was done. It's 20 years later, and we've learned a lot more in those 20 years. A lot more. So let's update the GIST-KIT connection to our present understanding. So GIST was originally treated or viewed as being just a single disease. But we now realize that GIST is really a kind of umbrella that covers a lot of different sarcomas, differing at the molecular and cellular level. Most GISTs, as was observed in 2000, most GISTs have a mutation in the KIT gene. But about a quarter of them do not. That is, they express the wild type form of KIT. They carry the normal gene. So when we say wild type, that's just genetic jargon to mean normal, the normal form of the KIT gene. So what are those 25%? You can think of this as a pie out of which we're cutting smaller and smaller slices. The biggest slice of the pie, 75%, is KIT-mutant. But about 25% of the pie is not KIT-mutant. A remaining 15% or so, these numbers differ a little bit from study to study, most of the remainder have a mutation in a closely related gene called PDGFR alpha or PDGFRA. Even smaller slices have mutations in other genes like RAS, NF1, and NTRK, and probably a few others that we're still chasing down. Very importantly, these less common KIT wild-type forms of GIST are biologically and medically distinct from KIT-mutant GIST. We believe that they are biologically different and require different treatment. Let me emphasize again that all of these forms of GIST are derived from ICC cells. They all, or at least 98, 99% of them, express the KIT protein, whether or not the KIT gene is mutated. Again, that shows us why the IHC test for the protein tells us that a tumor is a GIST, but we need to do additional testing, DNA mutational testing, to determine whether it's a KIT mutant GIST. Mutations matter. And this is a website recently put up by Blueprint Medicines. It's mutationmatters.com. And I recommend you take a look at that website. It gives you a lot more information about genetic testing and its importance in GIST. Mutationmatters.com. Okay, in the next part of the talk, I want to turn back to talking about the KIT protein and to tell you a little bit about protein structure and how that's relevant to KIT and to mutational testing. So I said earlier that proteins are large molecules. They're made up of hundreds or even thousands of amino acids in a particular sequence. It's often the case, and this is a bit of an oversimplification, but it's often true that a protein can be viewed as made up of substructures like Lego blocks stuck together, and each of those substructures we refer to as a domain of the protein. A domain might be, say, up to 100 amino acids long. Just as we think of a chain of nations, here's Central America, which is a chain of, leaving out El Salvador, I apologize, and Belize, but it's basically a chain of Guatemala linked to Honduras, linked to Nicaragua, and so on. And so you can think of the domains of the protein as being separate regions joined together in the way that different countries or states are joined together in a linear fashion. Each domain of a protein corresponds to a separate segment of the gene that codes for that protein. And those segments are called exons for reasons which we're not going to go into here. But the terminology is exon. The KIT gene has 21 exons. So as an analogy or metaphor here, we can think of the human genome as being a complete library of information. An individual type of protein like KIT represents one book in that library that contains maybe 30,000 books. You can think of an exon, and therefore a protein domain, as representing a chapter in the book. and an amino acid being an individual word or even letter in that chapter. This, and I do not expect you to memorize it, that's a joke. This is the KIT protein. I show this to you to emphasize just how much detailed knowledge we do have. So each letter, the KIT protein has 976 amino acids, or amino acid residues, as we say. Each amino acid, as I said earlier, is represented by one letter. So this is a chain of 976 letters. We just break it up into sections of 10 so we don't lose our position. And you see they're numbered from 1 to 976. And the wild type KIT protein is precisely this set of amino acids in this sequence. Any deviation from that sequence represents a change or a mutation. And we know, using genetic techniques that I'm not going to discuss here, we know where the exons are. There are 21 exons in the KIT protein. And if I just flip back and forth, you can see here's the complete sequence, and here it is broken up into the individual exons or domains. There are 21 of them. We know that from the gene studies. And I'll highlight two of them, exon 9 and exon, which I've highlighted in yellow, with two particular important residues in red, and exon 11 that I've highlighted in blue. So these are two of the 21 exons making up, or domains making up the KIT protein. Now, I'll spend a little bit of time on this slide. You've seen a version of this picture before. Here's another picture. What we're looking at are cartoons or representations of the KIT protein itself. KIT is a large protein, almost 1,000 amino acids. It is what we call a transmembrane protein. So every cell is surrounded by a lipid or fatty membrane that defines the volume of the cell. And the left picture that's shown in these yellow wiggly lines right in the middle, that's the lipid membrane of the cell. KIT is a transmembrane or crossing the membrane cell protein. Part of the protein resides inside the cell. Part of it resides outside and communicates with the external environment. And then by definition, part of that protein crosses the membrane, goes right through the membrane. We call that the transmembrane domain, as represented on the right-hand side. So we have an exterior or ectodomain of the protein. We have the interior domain. The interior region is where the kinase activity, the catalytic activity of the protein resides. And now if you look at the picture on the left, you'll see that exon 9 is close to the membrane on the outside. Exon 11 domain is close to the membrane on the inside. So we call exon 11 the juxtamembrane or close to the membrane domain. Most of the mutations in GIST, KIT mutations in GIST, occur in exon, initial mutations anyway, occur in exon 9 and exon 11. Why? Because it's mutations in that region that cause the protein to change its shape and to switch from its inactive to its active form that signals the cell to grow and divide. Okay, that's the end of my part A, the initial survey. And I guess I'll just go on to give you a little bit of deeper information, the part B of the talk, in which we'll give a little more depth about some of these topics. And I promise you that I'll stop in just a few minutes because we do want to keep on time. So let's say a little bit more in depth then about KIT mutations. KIT, as I said, is an oncogene. It's a gene which, when mutated, encodes a protein product that tells the cell to keep on dividing, whether it should or should not. Sometimes we refer to that as a stuck gas pedal, a gas pedal in the car that's stuck to the floor and keeps the car moving, whether it should, even if it's going through a red light. So the KIT gene, when it's mutated, acts as a driver that tells the GIST cells to proliferate. KIT protein is a particular kind of enzyme or protein catalyst called a tyrosine kinase. The details of that we won't go into here. It's a protein that acts on other proteins and triggers what we call a signal transduction cascade. The job of the drugs is to shut down KIT activity. That's the mechanism of action of imatinib and the other other GIST drugs. As I've said, in about 3/4 of GIST cases, the KIT gene is mutated and drives the tumor growth. It's true, though, that even in the remaining cases, perhaps even in wild-type GIST, even with the wild-type GIST, there may be some therapeutic benefit in inhibiting KIT. That's still not entirely resolved, but there certainly is in the KIT-mutant case. The KIT mutations in GIST are almost always, there's some very rare familial cases, but it's almost always a somatic mutation. That is a mutation that occurred in the patient's body during the lifetime of the patient, during development or adulthood. Therefore, the mutation does not affect the germ cells, the egg or the sperm. It cannot be passed on to a patient's children. There are rare, very, very rare exceptions of familial GIST, but almost always it's a somatic mutation. There's a great diversity of mutations in GIST. The GIST driver mutations can occur at many different sites in the KIT gene, affecting different sites in the KIT protein. And as we've mentioned already, sometimes GIST driver mutations occur in genes other than KIT, such as PDGFRA, that we'll hear more about later on. Critically, the site of the mutation affects the prognosis and the treatment. That's why we have declared more than 10 years ago as patient advocate organizations that mutation testing should be performed on all newly diagnosed GIST cases. Unfortunately, we haven't got to that point yet in most countries. Again, I'll refer to that Mutation Matters website that will give you more details about that. Here's another pie chart showing the different mutations that drive KIT. I've referred to this already. Most of them KIT, many of them KIT exon 11, a few in KIT exon 9, fewer in PDGFRA, and then tiny slices of the pie that aren't even shown here that are in other genes like BRAF and NTRK. KIT is one member of a large family of related proteins. PDGFRA is a closely related protein. So what we're looking at in this picture is really a family tree, showing you the relatedness of some of the many hundreds of different protein kinases in the human genome. And you'll notice that KIT and PDGFRA are very close in the family tree. They're closely related proteins. ABL, the protein targeted by imatinib, that is mutated in CML, that's a very distant cousin of KIT and PDGFRA. Again, the KIT protein with its 21 exons. Exons 9 and 11 are the regions where the primary mutations usually are found. PDGFRA is different but closely related evolutionarily, similar protein to KIT. And just like KIT, it's got an extracellular and an intracellular domain, and the exons are a little bit different in PDGFRA. So the exon corresponding to 11 in KIT corresponds to exon 12 in PDGFRA, for example. But they're very similar proteins. Last thing I'm going to talk about then is how do we understand mutation terminology? So you've heard reference already to PDGFRA D842V. People toss off those technical terminology very quickly. You might have heard, for example, about KIT V560D. What does that mean? Well, if we say KIT V560D, what we mean is that amino acid 560 out of the nearly 1,000 in the KIT protein, that 560th building block, has changed from the normal V, which stands for the amino acid valine, to a different residue, aspartic acid represented by D. That's KIT V560D. You may also have seen designations like W557-K558DEL. What does that mean? DEL stands for deletion. This is a mutation in which those two amino acids encoded on the code for those amino acids on the gene is simply missing. Those nucleotides in the DNA have disappeared, they've been cut out, and we have two amino acids missing, making the KIT protein 2 amino acids shorter, and those are critical amino acids. So I'll end at this point by just introducing the concept of targeted drugs for treating GIST, which we've heard talk about before and we'll hear talk about again. So the first targeted drug arose from the study of chronic myelogenous leukemia, or CML. That's a rare leukemia. Biologically, it's got absolutely nothing in common with GIST. It's a totally different kind of cancer. But it turned out remarkably that the driver mutations were related. The mutation causing CML is in the ABL gene, ABL. The mutation causing most GISTs is in the KIT gene. And it turns out, as we saw in the previous picture a few slides back, that in fact, ABL and KIT are related at the molecular level. They're both tyrosine kinases. And it turned out that some of the drugs, many of the drugs, like imatinib, that shut down ABL can also shut down KIT. And that's why the drugs, you had this unexpected benefit that the drugs developed for one cancer, CML, turned out to be very effective for KIT. Now, just to conclude, I'll point out that now in the last couple of years, with the help of drug companies like Blueprint and Decipher that are present at the meeting, we're now moving from the first generation of GIST drugs, all of which were developed for other cancers, other targets, like imatinib developed for CML. We're now moving into a new era where we have drugs that were specifically developed to treat KIT and PDGFRA and other specific driver genes in GIST. And that's really why we're entering a new era in terms of GIST treatment. So I'm going to leave it there and leave a few questions for a little bit of time for questions. Thanks.